Okay, I'm going to, oh, how about that? Yes, now you can hear, I see. I'm going to try not to be too repetitive, because we all want to get to the hands-on part, right? So that comes in the afternoons, and it's great to speak right before lunch, so that way everybody has their full attention on me, I guess. Is there a clock by any chance, so that I know? Ah, I see, got it.
Oh, yeah.
So just to get an idea, which you may have been polled before, so how many of you do computational structural biology as your main drive in research? Maybe half. How many do experimental structural biology and want to kind of complement that with computational stuff? Okay, maybe about the other half. Good. How many of you have used Python before? A lot of you. I mean, before this workshop? Yeah? Okay. Good. Then we're in good shape. So let's see. The idea with Proti, and let me do this, is that we have, these are folding simulations that are performed with molecular dynamics, okay, over the years. And these are all, as you can see, relatively small proteins that fold fairly quickly. So the villain headpiece is a well-known example of a fast folder and great for simulations. And as the years have gone on, the folding times of the proteins that can be simulated have gone up. And we can see that the folding times are going up exponentially as the years go on linearly. So this is what we might call Moore's law, right? What's happening is we're getting faster computers so we can fold proteins faster, so we can run simulations faster, but we're going to reach some limit. So there's a time scale limit that we can find actually just by doing molecular dynamics using all atoms. So there is a need for doing coarse-grained type of research. And something like Proti, or the elastic network model, can do that very, very quickly, so at least you can get an idea of how things move. On the other hand, we also have a lot of structural data coming from the PDB. This is from just about a year ago. It was reported in Nature that the PDB has hit 100,000 proteins. Now I think it's got about 110,000. So we're getting a lot of structures, and we want to make use of that, right, of all the structural data that we have. And the structures, it turns out, aren't just for different proteins. The structures are often the same protein that has that is bound in different states or has a different crystal structure. So you have the repeats of one protein over and over again found in the PDB. And maybe we can use that to find out something about that protein, the fact that we have it bound in so many different forms. And this is what Proti is for. It's just to exploit the PDB so that instead of doing these long, cumbersome simulations, you can use information that's already there in sort of an intelligent way and figure out some information about how these structures are moving around. And this is a slide very similar to one that Yvette showed, just saying that you start off with some input and then you can find out a lot of information very quickly from the PDB and get some idea about structural dynamics. I won't go back over that. Again, just nice pictures of what Proti is. This is also a year old. Proti is mostly Python. You can see 80% Python. There's some C in the back end. You don't see the C. You interface with it through Python. And then there are others. I don't know what the others are offhand, but there are other programming languages. It gets downloaded a lot, and it makes use of all of these different tools. IPython, which is what we recommend for all of these tutorial sessions. These NumPy or Matplotlib, so that you can view and you can do quick calculations, numerical calculations. And then you get all of these nice looking outputs that come out here. This is just so you can see that you can look at things. And once again, this is sort of an evolving suite of tools. It started with Proti. Now we're using this eval. I think this is growing more quickly than Proti these days. And this is to study the evolution of sequences and how that relates to structures. And then there's these two plugins relate to VMD, Drug GUI and NMWiz. And you already saw something about NMWiz. And Indira will talk about Drug GUI tomorrow. So just to go over the tutorials quickly so you have an idea of what to expect. That way you'll know what to do in the afternoon instead of having to spend the first half hour or hour looking through each tutorial and guessing which one is best for your research. The two real basic ones are the Proti tutorial and the structure analysis tutorial. The Proti tutorial just tells you what Proti is, introduces you to how to start a session in Proti and some of its capabilities. The structure analysis tells about getting things from the PDB and looking at structures, analyzing structures, aligning them, very simple types of procedures using structures. So the things that are found in this tutorial I think are going to appear in most of the other tutorials because this is telling how to interface with the PDB, and Yvette has already shown some of the functionality involved in these. Now the elastic network model tutorial. This is useful if you want to look at elastic network models, which already you've heard about. And there is a lot of capability that Proti has as far as using elastic network models that is not necessarily in the tutorial. So you should ask those of us who are around if you have any questions about things that might go beyond what's just in this tutorial. And I'll talk about some of the things that you can do with elastic network models beyond what's in the tutorial if you know a little bit of Python, which a lot of you do. So just to give an example. Okay, we've all seen, we just saw the elastic network model. I'll give my own little cartoon version. So here's, let's say that this is a protein with 10 residues. So it's a peptide. And we get rid of all of the side chains. We get rid of all of the backbone atoms except for the alpha carbon. So then we just say that we have 10 atoms that are separated by 3.8 angstroms. And we connect those to each other if they're close, so that this guy is connected to that one, and also along the backbone because adjacent residues are close to each other. And then we just look at how they move. So then you can get some little motion. And we analyze the slowest modes because those have the largest fluctuations. Somebody asked a question about the force constants. So I was listening and I threw this in here. So there are several ways of actually assigning force constants, and it basically doesn't matter which way you use. But don't take my word for it. You can look at it yourself. So the original, I guess, force constant was just using some cutoff distance. You say that all of these interactions that are put together when you build your network have a force constant of gamma if you're within some cutoff distance and 0 otherwise. So it's just a step function. And this is what's used in the ANM and the GNM. This is the default form of the force constant. You can change that in Proti because it's really, it's an API. You're typing everything into Python, so if you just know a little bit of Python, you can change that into, let's say, a form like this with an exponential decay, so that the force constant gets weaker as you get farther away, as the interactions get longer. So this is something that you might expect is more realistic. You're not saying that there's just a sharp step. Interestingly, if you do the dynamics, you find out that you get very similar dynamics. There was another, even more complicated form that's shown here that Conrad Hinson suggested. You can put that in if you want, or you can put in something like this, inverse power law type of decay, whatever you want. You can change force constants. And in fact, somebody else asked a question about different types of residues. So you can put in force constants for different types of residues. And there's an example of a study that we did where we did something very similar. And what we said is, how do we want to optimize the force constants? And we did this using the Gaussian network model by getting a lot of NMR structures from the PDB, calculating what are the fluctuations of all those residues, which you can do, you can fetch the PDBs, you can calculate mean squared fluctuations, and then assigning some topology to the elastic network model. And based on the fluctuations and on the topology, we can find out what are the force constants that are going to optimally reproduce the fluctuations that we look at. So here's just a little example. What we have here is I have colored interactions based on the separation along the chain of the two residues that are interacting. So k equals 1 means nearest neighbors along the chain. K equals 2 means that they're separated by one residue in between, and that will be in red. and then 3 and 4. And I've just plotted what are all of the force constants, optimized force constants, as a function of distance. What you can see is, of course, k equals 1 has all at 3.8 angstroms, and you get a sort of a large variance, but then there are different patterns. And if you look at trends, you can see that force constants for second neighbors have this sort of sinusoidal shape, and force constants for third neighbors have kind of an exponential decay, and you can go into details and look for interactions, look for specifics with residues, things like that, which we didn't find any. But
what is the inset on the left panel?
this one? This is, what is this? These are average. That's a good question. No, I'm sorry. This is showing, in fact, these two models. Something like this, and then the exponential decay. In fact, the best fits to what we find with the average force constants just as a function of distance to see whether or not either of those two is better than the other. And it turns out that both of the fits are good, that if you don't have just a sharp cutoff distance, that you do get kind of a decay with distance. Thank you for asking. Thanks for, it brings me back. In fact, this has been incorporated into Proti as gamma structure based. So we've just come up with a little bit of a contrived sort of parameterization of force constants based on the secondary structures, based on the separation along the chain of residues. And what we can find is that it gives just a tiny little increase over what the ENM would give, what the GNM would give when we're comparing it to NMR structures. So what I'm showing is that the black curves are showing fluctuations taken from NMR. The red curves are showing fluctuations taken from GNM, and then the blue ones are showing if we use this modified kind of force constant. And this is probably the best improvement shown here on the bottom. You can see that it's not really that significant. You get a little bit more of a bump in this loop region and a little bit more of a bump in this loop region over what the GNM would give. But the point here is that you're free to change force constants. You're not going to find that it makes all that much of a difference. But you can find out how to do that in the ENM tutorial. Moving on, the ensemble analysis tutorial. We'll tell about analyzing ensembles, which is also similar to what we've just done here, right? I just described looking at NMR ensembles and then fitting force constants to match the fluctuations that we see or that we infer from NMR ensembles. In this case, we might look at just ensembles in general and try to figure out how the protein is moving based on a collection of structures. And I get the fun, well, I can explain this, although I'm not an author. I'm sure that Yvette can explain it much better. She's an author on this paper, but I'll do my best. So an example is if you want to compare principal component analysis and an elastic network model results. This is, in the case here, HIV1 reverse transcriptase. And there are several structures in the PDB for this. So there are unbound structures, which are shown in red, that kind of populate this little area. There are inhibitor-bound structures, which are shown in blue, and then DNA-bound structures that are shown in green. And what has been done here is all of those structures were collected together and aligned on top of each other, and then principal components of the deformations or of the fluctuations, differences between those structures were calculated. And here, the structures themselves are plotted in the two-dimensional subspace made up of principal component 1 and principal component 2. So you can see that binding itself is going to make a large change along principal component 1 because the unbound structures have a large value on PC1, and those that are bound have a small value on PC1, and that binding to an inhibitor seems to have some effect on PC2. And these are just showing what those motions are. So the reverse transcriptase is sort of a hand type of structure where it has a palm and fingers and a thumb. And the first mode is motions of the fingers and the thumb. And the second is the motion of the thumb. And what we can see is that the ANM, the second mode of ANM, agrees almost perfectly with the first principal component of the structural ensemble. So this is really quite a striking correlation, I would think, between these two, which is just showing again that if you don't have an ensemble of structures, if you have a single structure, if you use an elastic network model, you can get an idea at least of what the principal components would be if you did have some ensemble of structures. And then similarly for PC2 and ANM3, the mode 3 of the ANM, you also get an agreement. And another example from the same paper showing the same thing with MAP kinase. What we can see is that, again, we have different structures, and they're scattered around on principal components 1 and 2. And again, we can see that the motions of the principal components, the low principal components, or the first ones, agree with the predictions made by the anisotropic network model very well. So once again, if you can calculate what these modes are using ANM, then you can get an idea of what you would find through an ensemble of structures. Or yes?
How many structures and how diverse do they need to be in order to be covered?
As many as possible, and as diverse as possible. So, right, in order to get an idea, you can use very few structures. So what actually happens mathematically is, yes? Okay, then 40. That's the number of modes that you get is going to depend on the number of structures that you have. So more structures will give you more modes up to the number of residues that are in the protein, or 3 times the number of residues that are in the protein. And then after that, what will happen is they'll just become more refined. You'll change the modes. So with trajectory analysis, I'll be honest, I haven't analyzed trajectories in Proti. That's just because I'm not really somebody who does a lot of molecular dynamics. This is for molecular dynamics trajectory analysis. You've all learned about MD recently. Proti is able to read DCD files and to analyze them to align structures that are in DCD files quickly. So if you try doing that in VMD, you'll find that it's substantially slower than what you get in Proti. So this is just a way, if you have long trajectories in particular, you might want to look at the trajectory analysis tutorial, because that will tell you something about how you can analyze it. You can also perform essential dynamics, which is just PCA, but you're using snapshots from the trajectory, right? So you use those as a structural ensemble, and then you can align them and calculate principal components based on that. Now, conformational sampling is also something that somebody mentioned about whether or not when you do ANM, if you're going to have collisions, steric clashes, when you deform the structure along some mode. And sometimes, yes, and I mean if you deform it too far, right? Because this is just an approximation saying that you have a crystal structure that's at the bottom of a potential well, and you're saying that your modes are tangent to some curved surface, let's say. So you have a multi-dimensional curved surface energy landscape, and the modes are showing you tangents to that. So if you extend one mode to a very large distance, the modes are straight in space, but the protein is constrained because it's a chain, right? So you have these backbone atoms are connected one to the other. So there's only so far that you can move along a structure without making it somehow energetically unreasonable. What the conformational sampling allows you to do is you can calculate a mode in ANM or using PCA, and then generate a bunch of conformations along that mode. And for each of those conformations, put it into NAMD and do an energy minimization. So that way, you're not sampling these unrealistic conformations that might come about because of the second order approximation that is in ANM. So in this case, let's say you use ANM. The strength of that is, of ANM is that you can come up with these large-scale motions. You come up with large-scale motions using these modes, and then you can sample those large-scale motions and energy minimize them and get an all-atom realistic conformation that comes from ANM and energy minimized structures. So this might also be useful. Now, druggability, as I mentioned, you'll hear about this tomorrow. I happen to like the druggability suite. This is a plugin to VMD. I like this even if you don't do drug discovery, you might find this useful for setting up MD simulations, which is not really what its intent was, but it works well because it just has a GUI interface for setting up molecular dynamics simulations. You can set up a whole slew of them that will each have a different seed. And then if you have access to a cluster, you can just run all of them simultaneously. Kind of an overview of what happens in druggability is that you have your molecule, and if you want to know whether or not that molecule can be affected by a drug, then what you want to do is look for places on the surface that it can bind. But the problem is that the molecule moves. It's not stationary, as you've already heard. So you do a simulation. And you do the simulation in the presence of probe molecules, for example, isopropanol, and look at where those probe molecules or how those probe molecules interact with the surface of the protein over the course of the simulation. So you do a simulation or several simulations, and you look at the distribution of your probe molecules during those simulations. You compare them to what you would expect just from having, if the molecule wasn't there, you know, what you would expect from chance. This is just statistical physics, I guess. And look at where these probes are, or how they are distributed over the surface of the molecule. And if they're found to be in certain places for a long time, then you can say, well, you know, right here we might have some binding site because the probes are populating this area with a higher than random chance and a significantly higher than random probability. And then we can say, well, what kind of probes? Let's look at their chemical properties. Are they positive? Are they negative? And so on. So this you will hear more about tomorrow. And eval. So the evolutionary properties. Who will be talking about eval? Jihan will be telling you all about this. So he's probably our main programmer these days. And I will try to do justice to eval right here. So an interesting aspect of protein dynamics is that it is coupled with evolution. And we can look at that in a very simple way. These bars are showing sequence entropy. And by sequence entropy, I mean how stable a particular area in a sequence is. So how stable a residue is against mutations. Those low entropy areas are showing that, shouldn't it be the opposite way? No, this is fine. Okay, the low entropy areas are showing that you have very low mutation rates. High entropy areas are showing that you have a high probability of basically any residue occupying that state. So if you have high entropy, you don't know which residue is going to be on that site. If you have low entropy, you know very well which residue is going to be on that site. Yes? How is entropy calculated? Oh, look at the source code. No, so in this case, what happens is we take a multiple sequence alignment, and then we can just count what is the probability of finding different residues at that part in the sequence.
Is it like counting tons of versus the others?
Yes. 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 So for each residue, we can find so there are 20 different residues, or maybe 21 if you include gaps, and then we can assign probabilities for each of those. And then if you sum over the probability for all of these 21 possibilities, negative p log p will give you an entropy. This is just one measure of how conserved a region is in a sequence. Okay, so yes, so tomorrow Yvette will tell you more about this. I'm just overviewing. And in fact, the eval tutorial is scheduled for tomorrow if you want to look at that. We can overlay on this the mobility that we get, let's say, from looking at all of the modes, which would be this black line. And what we will find is if we plot how well a residue is conserved and we plot its mobility, what we find is that there's agreement. So we will see that there are conserved residues, let's say on the inside of the protein, where it's necessary to keep some kind of an interaction to keep the protein together. And that turns out to be site of low mobility. And then you can get a lot of variation in highly mobile sites, right? So residues that move around a lot tend to mutate a lot. And we can look at mutual information, which is just saying how much, how often are two residues co-mutated. And what we can find is that we have certain incidents which are off the diagonal. You might expect that you see residues that are co-mutated if they're close to each other. We can find certain incidents of residues that are co-mutated that are off the diagonal that have high probabilities for being co-mutated. So in some cases, they might be that you have a disulfide bridge, which would be this little circle right here. We've got a disulfide bond, or a salt bridge, which would be this little circle over here. And in other cases, we can find that there are residues that are implicated in the transmission of allosteric signals, which might be these ones here. So you'll hear more about this tomorrow. This is just comparing structural dynamics to mutation rates. Interestingly, this can be used, this is being used increasingly to predict structures for proteins where you only have sequence information. Because if you know how things are mutating and co-evolving, then you might be able to say that these are residues that are in contact. So something to keep in mind. Normal mode wizard, I was going to show you that. I think you just saw it though. So this I also think is a lot of fun. We have several tools for calculating the modes from A&M. In fact, there's a web server that's hosted at Pitt that does this. I really like to do this in VMD, just fire up VMD and use the normal mode wizard because you can rotate the molecules and it does everything very quickly. This is some application of Proti that just works right out of the box. So I want to spend some time discussing what else you can do with it. So there are these tutorials which go over really, the basic things, but the point is if you know a little bit of Python and if you know how to use Proti, then you can do a lot more than simply align one ensemble and look at how these motions are. So I'll give you a few stories. In this case, this is also something that Yvette mentioned. This is a protein, don't know which protein. We can calculate, and I should know what this protein is. We can calculate the modes for this protein. We can look at what we would expect just looking at fluctuations from A and M. And we would get something like this, right? Some little trace with a big spike here. And the spike comes from this tail. And it's known as the tip effect. This is a well-known issue that crops up when you're using elastic network models. And this comes because these residues on the end are not very highly connected to the rest of the protein. So you're going to have, let's say, lower values on the diagonal of the Hessian in this area, or lower connectivity. So these residues are more free to move around. And therefore, they're going to have large fluctuations. But what we see when we compare it to the B factor, does everybody know what a B factor is? I think so, I hope so, okay. When we compare it to the B factor is that there's not really that good agreement. And part of this is because you've got this huge tip effect, and there might be some bumps that actually correlate with the B factor, but this is changing the whole scale. So why might this be? As was mentioned, it might be because of the crystal. So we can say, what about the crystal contacts? And how do we do that? With Proti, we can, if we enter in the crystal structure and we wanted to look just at one in the middle, at one structure in the middle, we can build a Hessian for the entire crystal in the region of this one protein, just like this. And then what we can do is we can say that the one in the middle is what we will call the system that we're interested in, and the rest of it is just some environment that surrounds the system that we're interested in. And we don't really care about the environment except for its effect on our system of interest. So doing a little bit of math, what we can do is integrate out all of the degrees of freedom in this Hessian except for what is left in this system. And we can get an effective Hessian for this protein in the presence of the rest of the crystal. And this is done with a command called reduce model in Proti. And you can find out more about this from the elastic network models tutorial. So if we do that, if we're looking at the crystal context, then we can see that it reduces this peak a little bit. It raises sort of the motion over here a little bit, and it helps. It turns out it doesn't tell the whole story. We can say that maybe the whole crystal is moving. If we say that the whole crystal is moving, then we can get much better agreement with the B factors. But this you can't do just using Proti commands by themselves. You just need a little bit of Python, and you can do that without too much of a problem. Another example is looking at global transitions, in this case of a membrane protein. So I'll tell the story. This is a glutamate transporter, and it's a trimer, a homotrimer, that sits in the membrane kind of like this. This transports glutamate, which is a neurotransmitter. And for a long time, only this outward-facing structure was shown. So this is outside the membrane, this is inside the membrane, and this is what we call the outward-facing structure. So the protein has a scaffold region that's in the middle, and then it has these transport domains that are shown in blue on the left and shown in magenta on the right. And after some time of knowing the outward-facing structure, an inward-facing structure was put on the PDB. And what it showed is that these transport domains move through the membrane. And this is really a large motion. If you align just the assembly domains in the middle, these wheat-colored things, and then you look at how the transport domains move, they make this dramatic motion like an elevator going through the membrane. And here's a, oh, sorry. So this is just to show what that motion will look like. The transport domain is shown in blue and yellow and red, and the assembly domain is shown in wheat, and the lines are showing where the membrane is. And so you can see that just making some model that we get this motion going through the membrane. I mean, this is simply an interpolation. This is just saying that we're going to take the outward-facing structure and just morph it into the inward-facing structure, right? But we didn't have a model that would explain that. And elastic network models didn't quite do that. Why is this one here? I'll get back to that, in fact. So the A&M modes, I can show you right here. If you just do an A&M in Proti, then you'll get some modes that look like this. Right? So what you see is that the trimer domains, that the transport domains are moving in and out in this radial motion, opening and closing. This is the third mode. It's symmetric. The first two modes are an asymmetric version of this one. But you see that they're not moving through the membrane as you might expect. And what we said was, well, maybe we can explain that. And we might be able to explain that by modifying the Hessian a little bit. So normally, an element of the Hessian, a 3x3 sub-block of the Hessian, or sub-Hessian, is going to be based on the x, y, and z differences between two residues. So we're looking at residues I and j, and then we have the changes in x, y, and z between I and j in the crystal structure. And we say that there's this force constant, gamma, that goes outside. So we very artificially said, well, let's take this force constant, let's move it inside the matrix, and let's attach it to each one of these components that is in there. So that way we have to take a square root. And let's even make it different for the x, y, and z components. So we make the x and the y components very strong so that the membrane is constraining the protein, assuming that in this picture we are looking down the z-axis. So we say that motions in the x and the y direction are going to be difficult, but motion in the z direction is going to be easy. And we just wind up with some modified Hessian that has different values on the z-axis or in the z direction than it does in the x and y direction. Okay, and if we do that, we can do this, in fact, with what we call build Hessian. If we do that, then the motions that we had before, again, We see that there's this large opening and closing motion, kind of a grabbing motion in A and M. Okay, that's one way to do it. If we modify the Hessian in this way, then we see that there's not as much of a dramatic motion. But when we turn it sideways, we get very much this up and down lift-like motion of all the subunits. And again, this is the third mode. The first two modes are asymmetric versions of this mode. And we find something similar for the inward-facing, if we start at the inward-facing structure and then go to the outward-facing.
So presumably, did you do that just in the residues that are within the membrane?
You can do that just in the residues that are in the membrane. In this particular case, it was done in residues that are all, for all the residues. And this is implemented in fact, it's in Proti in this IM, A&M class. Now, I want to show something. I'll show this movie again. So when you look at this moving sideways, you can see that there are these helices that run parallel to the membrane normal, or normal to the membrane, right? So you have transmembrane helices. And if you just make a modification like this, what's going to happen is those helices are going to want to stretch out like springs, which is, we know, not realistic because the helices are hydrogen bonded, so they retain their structure in reality. So what we did with this, let me go back now, is we used a method called rotations and translations of blocks, or RTB. Yes?
A quick question on that motion. Is that elevator motion seem very rigid? That assessment?
That it's a rigid motion?
Yeah, there was no internal motion.
Yeah, so this is exactly the point that I'm making. Okay, the question is that that motion seemed rigid. Yes, so there wasn't a lot of internal motion. And the reason is that for that particular model, what we did is we looked at the secondary structures in the protein, and we said each secondary structure, as defined by the DSSP, is a rigid block by itself, which means that each helix is going to remain as a helix. It's going to be a rigid block. And what does that mean? It means that residues within the helix don't change their positions relative to one another. So the block has 6 degrees of freedom. It can translate in X, Y, and Z directions, and it can rotate around 3 axes. And this is done numerically, not that it really matters, by taking this whole Hessian, and then we make a projection matrix into a space of rigid blocks with 6 degrees of freedom each. And we get a smaller Hessian that we diagonalize. And then when we get the modes in this smaller space, we project them back into the space of all the atoms, or all the residues. Yes?
Those conditions that elevated, they are in touch with membrane, right?
Yes.
And they are constrained.
They're not, yeah,
In
they're,
the membrane.
the idea is, yes, they're constrained by the membrane.
Then for me, maybe I'm right. It doesn't make sense that they can move that much up and down along with the very long organization of the membrane.
Yes, so that was the whole idea behind this, is that, why would the inward-facing structure not be very well explained by just what we find with the A&M? And the hypothesis was that the inward-facing structure, that the A&M allows complete isotropy of any external environment. And what we thought was that maybe this isn't the case, that the membrane is in fact restricting the radial motions that are intrinsic to the molecule. So we just increased the energetic cost of moving radially outward. So we are promoting, in fact, in a somewhat artificial way, but we're promoting this motion. And what we find is that it agrees very well with the motion that is observed when we compare the outward-facing and the inward-facing structures. And in order to get this to work properly, we use this rotation and translations of blocks, in which case I'll show the movie one more time because I love showing the movie. Now we're saying that every helix itself is rigid, right? So the loops are flexible, the helices are rigid. So if you look at one helix, it's not going to move relative to itself. But all of the loops connecting the helices are rigid. You can see that there are these little flapping motions in the loops. You can see that the motions that we're finding are in the loop regions. This is one way of preventing deformations of structures, right? Another way is sometimes you can just remove something. If you have, for example, some tail, the tail, because of either the N or C terminus, is kind of hanging off the protein. Sometimes that's cut off. Instead of cutting it off, you can just make it into some rigid structure, and that way you're not going to necessarily see such large motions coming from that, or large deformations. We also have, and this is partially implemented in Proti, we can do the same thing using explicit membranes. What we can do is fill in this case, let me put my pointer. We can just build a little chunk of membrane around the protein. So here's a side view, here's a top view, and we just artificially construct some membrane, which is just some lattice of particles that are supposed to represent the membrane. And we build an elastic network model containing this whole system. And then we integrate out all of the motion of the surrounding membrane. So we're left with just the motion of this protein. We find very similar results with that one. That, in fact, the motion, the radial motion of the protein that is intrinsic, if it's in an isotropic environment, will be repressed by the membrane. And then you get this up and down kind of motion. So that's a little bit more realistic. And this can be done. We have something for building the membrane. This is done essentially with reduced model. And there's now, although it doesn't say here, there's an EX-A&M class in Proti for looking at proteins in the presence of membrane. But wait, there's more. So you don't have to stop just at proteins. And even in Proti, you don't have to stop at just proteins. So another way of using elastic network models is to look at structures that can't be modeled because they're too big to be modeled using MD. So things that, for example, this nuclear pore complex in yeast. This is made up of over 400 proteins. What we're looking at here is each little blob is a protein. And then you have this nuclear pore complex that is put inside the nuclear lamina that allows things to pass into and out of the nucleus. But we wanted to look at these intrinsic motions of this because it's known that, for example, some viral capsids can pass through into the nucleus intact. And how does the motion of the NPC facilitate that? So this is something else that you can do with an elastic network model, is just model some huge structure like this where each little sphere is a protein. And then we can model just a little section of the nuclear lamina around it, and then look at how this is going to move. So what we can do is use gamma variable cutoff, which is a function in Proti that allows us to choose a force constant that has a cutoff distance based on the type of residue that we're looking at. So if you say that some residues have long-range interactions, or in this case some proteins, and some have short-range interactions, then you can use gamma variable cutoff to tell which residue has which type of interaction, or what is the cutoff for each. And alternately, you can enter in your own Hessian, and Proti allows you to parse a sparse matrix, to enter in your own Hessian and calculate modes by yourself. It will calculate modes using NumPy. So we can just look at what these modes are for some structure of this sort. We see that inside the nucleus we get large motions, outside the nucleus we get smaller motions. That's just some example. There are various commands that I went over. Those are the tutorials, just kind of a brief thing. If you know Python, and if you can use Proti, you can do a lot of different things beyond simply looking at proteins. You know, it's up to whatever you can imagine. So let me see. I just want to make sure that I have this. Yes. So there is a tutorial for this implicit membrane A&M. It's not with the rest of the tutorials. I put a link to it on this talk, but of course you don't have the talk, so that will go up at some point. This is on my own web page, kind of hidden. And we can see even that you get these up and down motions. All the arrows are pointing up and down. If you're interested in that, it will be hard to find. Ask me. I'll try to make sure that there's a link somewhere on the workshop webpage so that you can find this. And that's all I have. I'm early. Are there questions? Yes?
Thinking about the connection between the protein dynamics and function, do you find some motion or some modes are useful, but some are useless?
Well, we find that there are some modes that we can associate with known uses and some that we haven't yet. You know, I think Emerson said that a weed is a plant whose use hasn't been discovered yet, right?
Okay.
So,
just wonder what you find. So for the modes that are productive, typically how many? I don't know whether proteins they just use one mode or two to do the job. How many modes are typically used in the actual?
right. So how many modes are typically used? Typically one. And what we tend to, let me go back to the example that I showed earlier.
What I have in mind is, ATPs are used in a lot of the, actions. I was kind of think they bump the protein from one mode to another because it's a big kind of a kick from ATP.
Right. So, yeah, yes. So you might think, yeah, that ATP is just adding some energy. So if we assume that the energy that is coming into the protein is distributed equally among the modes, which I think is a reasonable assumption, then this is why we look at the slow modes. And what we find is that the slowest modes, usually just one, sometimes two in this case, will correspond with known motions. It doesn't mean that other ones are useless. And it also doesn't mean that those modes are telling everything about the motion, because there are these fine details that are taking place that aren't captured by the global modes. So all that we're looking at here are how the protein can move just in some equilibrium environment, right? When it's just being bombarded here and there, or if you just give it energy randomly, how is it going to move? It doesn't mean that there aren't some subtle interactions taking place between side chains. Those, of course, are important. You know, if you don't have the right chemistry and a binding site, then you're not going to have any function of the protein. But that isn't necessarily captured by these coarse-grained models. That's something. What I would like to say is that you use these large-scale motions to look at, or use coarse-grained models to look at large-scale motions, and then after that, after you have some idea, then you can use MD to look at what's happening on a finer level. And then, of course, still you don't know about these things like catalysis, right? Because that's too fine. So, did that answer the question? Kind of.
So, if you're curious which modes will be populated, can you just check which ones will be frozen out at room temperature? And also, can you just check PC from your PC and the ensemble structures, which ones are actually?
I'm sorry, if you're curious, which modes will be populated?
Yeah, if you're curious which modes are important, can you look at the PCA of the texture structures?
Yes. Yes. So.
Check to see which ones are frozen out at room temperature.
Are frozen out at room temperature?
Yeah. So some of them are going to be populated at room temperature.
Yeah, but okay. So if you're using an NMR ensemble, I guess, I mean, you're talking about solution NMR, right? Room temperature. Most of the structural information we have comes from X-rays, which are, not necessarily showing the biological state of the molecule. Also, in many cases, we don't have an ensemble of structures. So yes, if you have an ensemble of structures, I think that it's a good idea to look at what is that ensemble. And I don't know whether you should believe an elastic network model or a structural ensemble more. I guess that's up to your own preference in that. I guess it depends on who came up with the structure, right? Yeah, but if you only have one structure, then you can get a good idea of how it's going to move based on using an elastic network model. And this is what something like this is showing. When we do have an ensemble of structures, then that ensemble agrees with the elastic network models. But there are many proteins for which we only have one structure. In the case of this glutamate transporter, we had two. And before we had the second one, even now, I'm not sure how it moves because, before we had this inward-facing structure, there were E&M results that agreed with other motions. And we still have kind of contradicting things. We don't know how it works, but what we want to do is be able to build models that tell the story that agrees with the data. Yes?
Can you go over again the part where when you introduce the membrane around the protein, how do you take into consideration the interactions, like, I don't know, maybe some kind of hindering effect to the protein motion?
Yeah, so, and this is, okay, the membrane around the protein. The way that this works is we simply put the membrane around the protein. We build a lattice, let's say. And then anytime that we have one of our lattice points, which are shown as these green dots, love this laser pointer. Anytime we have one of these lattice points that comes too close to a residue in the protein, we just don't put it there so that we don't get any steric interactions. And then in this particular case, for this study, we used a force constant that was, so one point is the lattice of the membrane should have approximately the same connectivity as the protein itself. That is, if we look at the average number of links or of edges per node in the protein, that will be some number, let's say 20. Then we want to make the lattice so that if we use the same cutoff, we're going to get the same number of nodes for one lattice site on average. The second thing is that, so that's how that site is found. The force constant inside the lattice is about twice the force constant of what was inside the protein. So this we found empirically, basically, is because this is a coarse-grained model. We have really no idea of how a membrane is going to work. This is, if you talk to somebody who studies membranes, and I have, they don't necessarily agree with this, that you can do this, because membranes behave differently in the X and Y direction than they do in the Z direction. I mean, they're not just elastic nets. So we had to come up with, we had to adjust these things a little bit. We made force constants between lattice sites in the membrane, twice as strong as those in the protein, and then we use the geometric mean for the ones between the protein and the lattice. And I can talk to you about this more offline. Yes?
Question about the figure you were actually just at before, where you're comparing the PCA to the
Yes.
ANM.
This isn't the best way to, oh, no. This one.
Yeah. So on the bottom right, when you're using an ANM to generate a bunch of different modes.
Yes.
Which of, so there's like, what, 50 to 100 different structures that are being input. How do you
Yes.
choose just one from which to generate your different modes?
One structure from which?
Yeah.
Yeah, you choose the one that's closest to the mean of the ensemble
Okay.
as the representative.
Okay. Gotcha.
So you align everything, and then you figure out what is the distance from that from the mean, and use that as your representative structure. That's a good question.
Something in the Proti that sort of helps you, that runs that through and then shows that to you, or do you sort of do it by eye?
Well, yes, in Proti you can do that. There's something, when you make an ensemble, and this is in the ensemble tutorial, there's something called there's Git RMSDs. So if you have an ensemble of 1,000 structures and you do ensemble, if you iterate through the coordinate sets and then get the RMSDs, it will return those. And then you just have a list, a Python object that's a list. This is why it's nice to know a little bit of Python. And you can search through that for the min. And in fact, there's a Python function for getting the min. So you can just return which one in that ensemble has the minimum RMSD. All right, we're good. So the afternoon is for tutorials. And there are several of us around who will help with that. I don't know if there's any food yet, so I can't promise that.